The next plenary talk speaker is Pietro Valdastri. Professor Valdastri is a full professor and chair in robotics and autonomous systems at the University of Leeds. He directs the science and technologies of robotics and in medicine lab at University of Leeds. He received his laureate degree in electronic engineering from the University of Pisa in 2001 and his PhD in biomedical engineering from Scuola Superiore Sant'Anna in 2006. After his PhD, he became a professor at Scuola Superiore Sant'Anna. But in 2011, he moved to Vanderbilt University in the US as an assistant professor. And then he returned in 2006 to the UK to work at University of Leeds. Professor Valdastri has published over 100 journal papers in the field of medical robotics and has been the principal investigator of a number of grants in the US, in the UK, as well as European Union. Now he's won a number of awards, including the NSF Career Award, the KUKA Innovation Awards. Professor Valdastri is a senior member of IEEE, senior editor for medical and rehabilitation robotics of the IEEE Robotics and Automation Letter, a member of the steering committee of ISIMT. His research has been featured in the media widely, and he's also had some success in technology transfer by founding a company in 2009 that's been acquired by a larger enterprise in 2017. So both, in the industry as well as in academic, Professor Valdastri has enjoyed great success. So it's my great pleasure to introduce Professor Valdastri to give a talk on medical capsule robots, a fantastic voyage. So Professor Valdastri, please go ahead.
Thank you very much. Just to check, can you hear me fine?
Yes, yeah.
Yes, very good.
We can, yeah.
Very good, yeah. Thanks a lot for the kind introduction and for inviting me to give a plenary talk at ICRA. And welcome to everyone listening today. Before I begin, let me warn you that this presentation contains some scenes from preclinical experiments showing tissue, blood, and other things that some viewer may find disturbing. So viewer discretion is advised. But let me start now. When I was an early career researcher, I was always fascinated by talks given by Paolo Dario from Scuola Superiore Sant'Anna. He was my PhD supervisor and a pioneer of robotic endoscopy. He often mentioned science fiction as a source of inspiration, and in particular, he was used to refer to a 1966 movie called in this movie, a submarine crew are shrunk to microscopic size and venture into the body of an injured scientist to repair damage to the brain. Another example along the same line is the episode of The Jetsons, aired in 1962, where George Jackson goes in for a physical examination and the doctor uses a special capsule called Peekaboo Proper. Fast forward 38 years, to the beginning of the millennium, and this vision became a reality thanks to Given Imaging, which is now part of Medtronic. Given Imaging introduced into clinical practice the PillCam. This is a wireless camera integrated into a capsule that, once swallowed by the patient, is able to travel throughout the whole gastrointestinal tract, propelled by natural peristalsis while sending pictures to a set of body-worn antennae. This technology has been used by more than 3.5 million patients around the globe and is now the gold standard for the diagnosis of diseases in the small intestine. Of course, this technology also has a limitation, and in particular, it is not able to interact with the surrounding tissue and carry out intervention. These limitations sparked the brand new field of research within the robotic community, medical capsule robots. As robotics may provide controlled locomotion, advanced sensing, interaction with the surrounding tissue to perform surgery, and many other functionalities. So over the last 2 decades, a large number of medical capsule robots were developed by different research labs and addressing a variety of clinical needs and anatomical districts, from drug delivery to random biopsy, from legged locomotion inside the colon to underwater navigation in the stomach, and from in situ histology to local delivery of surgical clips. In my presentation today, I will share with you my personal perspective on how the field has evolved in the past 20 years, trying to highlight what has been accomplished, what have failed, and what were the lessons learned. But first, let's discuss the clinical motivation. So why medical capsule robots are important. Depending on their size, capsule robots can be designed to operate in different districts of the human body. However, their most common application has been in the gastrointestinal tract due to the relatively easy access via natural orifices and the huge benefit they can provide there. From an anatomical perspective, the gastrointestinal tract that you see here in this slide is a long tube composed by the esophagus, the stomach, the small intestine, and the large intestine here, or colon. And it is home to different diseases, including cancer. Looking at 2020 cancer deaths worldwide, we notice that gastrointestinal cancer combined account to more than 2.2 million and are the main cause of cancer-related deaths. If detected early, however, cancer can be treated effectively. Here, the example of colorectal cancer. As other cancer, it evolves in 5 to 10 years from an asymptomatic stage, where simple treatment translates in a very high 5-year survival rate, to a symptomatic stage, where the 5-year survival rate drops dramatically, and there are very limited options. For this reason, colorectal cancer screening is a common practice around the world, with screening programs starting as early as 45 in the US and Australia and 50 in the UK. The best way to screen for colorectal cancer is by colonoscopy, using a flexible endoscope that you see here. This is a 1.5 meter long tube with a camera here at the tip and illumination, and an irrigation channel to clean the camera or to insufflate the collapsed lumen, allowing for visualization, as you see here. The tip can be articulated in 2 directions by cable steering, with the cables at the tip connected directly to 2 knobs controlled by the thumb of the operator. The flexible endoscope also has an instrument channel to allow access for endoscopic instruments to interact with the tissue and collect biopsy for further lab examination, as you see here, or remove polyps, which are precursors of colorectal cancer. A very similar instrument is used to access the esophagus, the stomach, and the small intestine from the mouth in upper gastrointestinal endoscopy. It is interesting to note that the design of the instrument has not really evolved for the past 60 years. Since 1957, main progress has occurred in the visualization component, going from a bundle of optical fibers with the operator having to look through it, to ultra-high definition camera chips integrated at the tip of the instrument. All the rest has not changed. This out-of-date design is associated to all the downsides of flexible endoscopy. The first being pain. The flexible endoscope is designed to be introduced from its proximal end, and as such, the distal part needs to interact with the surrounding tissue to advance, as you see in the video here. So when a band is approached, the instrument must, by design, stretch the tissue to progress forward. This causes pain in the patient and increases the risk of perforation. Then the user interface is highly unintuitive, with the thumb controlling 2 degrees of freedom and the other hand used to move the endoscope forward and backward. And finally, the instrument is extremely expensive by design, with all its integrated components. So flexible endoscopes must be reprocessed after each use, increasing the risk of cross-contamination and limiting the availability of procedures to center properly equipped with cleaning facilities. Medical capsule robots have the potential to provide a solution to all these problems, offering a painless and less invasive experience, using robotics to facilitate or even automate the procedure, and possibly reducing costs, enabling single-use devices and procedures that can be offered outside specialized facilities. So what are the components required for medical capsule robot to achieve these goals. They must possess proper actuators and mechanisms to interact with the surrounding anatomy and move around in a controllable fashion. They need sensors, including vision, to enable clinical diagnosis and other functionalities. They need a brain or a CPU, communication with the outside world and the operator, and finally, they need power. All of this needs to be integrated in a limited volume, so size becomes the most important cross-cutting constraint. Typical target values for a capsule robot are a diameter of 11 millimeter and a length of 26 millimeter to allow for oral intake, or a little larger volume, typically 3 by 3 cubic centimeters, for application in the colon via retrograde access. Of all of these components, probably the most interesting for our community is the part related to the actuation. So I'll focus on this in the next slides, but I'm more than happy to discuss the others after the presentation. The first approach was to adopt onboard actuation by actively controlling the capsule using internal miniature locomotion mechanisms. Given the constraint in size, the most effective way to explore onboard actuation was to focus on the specific needs of the different anatomical districts. For example, the esophagus is a tubular cavity that is relatively easy to access. There, the main need is to slow down or even stop the passage of a capsule robot, which would otherwise take a few seconds to reach the stomach. The stomach is a large collapsed cavity that needs three-dimensional motion capabilities. The small intestine is a long tubular cavity that is pretty difficult to access as it is located in the middle of the gastrointestinal tract, and its diameter is about 1 centimeter and is the same as the given imaging PILCAM that we discussed earlier. And finally, the colon is a tubular collapsed cavity about 3 times larger than the small intestine. There, a medical capsule robot needs to create space, move forward and backward with a reasonable speed, and have the possibility to interact with the tissue to get samples and remove lesions. Now, for the sake of time, I will only focus on stomach and colon, where the incidence of cancer is the highest. But it is also worth mentioning that the small intestine is served very well by the clinically available wireless capsule endoscopes like the PILCAM, and the esophagus is extremely easy to reach via flexible endoscopy. As for the stomach, the first issue is to create space. So while gas insufflation is typically used in flexible endoscopy, an alternative is asking the patient to drink about 1 liter of clear liquid to expand the stomach. To support this approach, within the European project Vector running from 2006 to 2011, and coordinated by Mark Schur at Novineon and Paolo Dario at Scuola Superiore Sant'Anna, a volunteer drank a liter of water and then swallowed a PILCAM. The volunteer was then asked to assume different positions to allow the capsule to visualize different anatomical landmarks in the stomach, demonstrating images of acceptable quality to perform a diagnosis. So this experiment showing that water or clear liquid is an acceptable way to expand the stomach underpinned the development back in 2009 of a 3D printed 4-propeller miniature submarine, integrating 4 DC motors, a lithium-ion polymer battery, and wireless microcontroller, all with a diameter of 11 millimeter and a length that was less than 3 centimeter, with additional space to accommodate a camera. Here you can see different videos demonstrating the technology, with the capsule performing a figure-8 trajectories at different speeds and dexterity trials. While this prototype was battery operated, thus with a limited lifetime, we also explored, in collaboration with Bob Puer's group in Leuven, wireless powering. We did so by integrating a set of 3 perpendicular coils and transferring power with the carrier at 1 megahertz from an external coil to be placed around the patient. In this experiment, we were able to transfer an average power of 400 milliwatt and operate successfully all the 4 propellers, all within the safety limits allowed for human exposure to electromagnetic fields. Well, after demonstrating the feasibility of this approach, we did not pursue it further. It is, to me, really exciting to see recent announcement from a Silicon Valley startup that is trying to bring a 4-propeller-based capsule robot to the patient for non-invasive inspection of the stomach. This company is called Endiatics and have been successful in demonstrating the technology in humans in 2020. You can even find the video on YouTube with one of the founders swallowing the capsule robot and streaming images directly from his stomach. So the idea of swallowing a small submarine to get a diagnostic test may soon become a reality. Now, let's focus on the colon. There, an intriguing way to create space and at the same time propel the capsule robot forward and backward has been legged locomotion. Legged locomotion was pioneered by Paolo Dario and Arianna Menciassi's group at Scuola Superiore Sant'Anna in the first decade of the millennium via multiple projects, including Hemilock, Optimus, and Vector. Prototypes of increasing complexity were developed using shape memory alloy actuation first, and then moving to miniaturized electromagnetic brushless motors. The number of legs increased from 4 to 4 plus a balloon, to 8, and finally to 12, to provide a more uniform lumen distension and a more efficient propulsion. In this version, two brushless motors controlled independently 2 sets of 6 legs. Depending on the gate of actuation of the motors, this capsule was able to propel either forward or backward at a speed of 5 centimeters per minute, generating a pulling force of 3.8 Newton. While this robot was an interesting example of mechanical design and miniaturization, legged locomotion was flawed by several limitations. The first was complexity. The capsule was made out of 72 miniature components, some requiring multiple technologies for their fabrication. As you may imagine, assembly was not that easy either. This meant a very high cost of production, thus a very unlikely translation into a cost-effective product. The second limitation was related to power requirements. In order to complete its journey along a column of average length, the capsule required a battery pack that was about the same size of the capsule itself. And this was clearly unacceptable from a clinical standpoint, as ingesting 2 interconnected capsules would pose a high risk of retention. So due to complexity, powering, and size constraints, we had to abandon legged locomotion and get back to square one in search for a more feasible actuation strategy. Then, while scratching our heads in lengthy brainstorming meetings back in 2009, we came across this science fiction clip. Here, a permanent magnet is used to retrieve several different objects from the upper gastrointestinal tract of the subject. And then the big idea. Why don't we use magnetic manipulation? So, placing a permanent magnet inside the capsule robot would enable an external magnetic field to generate forces that are related to the applied field gradient and torques that are related to the misalignment of the 2 fields. So by controlling the external magnetic field and field gradient, we are able to apply a controlled wrench to the capsule robot without the need for storing power or integrating complex mechanisms on board. Well, magnetic manipulation has been explored since 1950 to guide cardiovascular catheters with a magnet integrated at the tip. The first example of magnetic capsule robots dates back 2008, when within the European project Vector and in collaboration with Ovesco, we designed a capsule for the controlled deployment of a surgical clip to stop bleedings in the gastrointestinal tract. This here is the over-the-scope clip, typically released by a flexible endoscope, as you can see here. And this is the capsule robot that we designed, integrating a mechanism to release a clip when receiving a wireless command from outside the body of the patient. The release mechanism is actuated by a brushless motor integrated on board. A permanent magnet is also included in the capsule, allowing for remote control and manipulation. In this in vivo experiment, the capsule robot is moved toward the bleeding via magnetic manipulation. Then, once the clip is in position, facing the bleeding, the operator sends the wireless command and the clip is released. Then the capsule is moved away, still by magnetic manipulation. In this first example, the external permanent magnet was supported by a passive arm and manipulated directly by the operator, often limiting the precision of motion. To improve this, we mounted the external permanent magnet on a robotic arm. As you can see from this video, using a small cylindrical permanent magnets in the capsule enable a very good level of control over pan and tilt. Then we repeated the experiment using an in vivo animal model and comparing robotic against manual control of the external magnet to manipulate the capsule inside the colon. The results were clearly in favor of robotic control, so we decided to adopt this strategy in all our research platforms. Of course, using a permanent magnet outside the patient is not the only way to generate the driving magnetic field. Another possibility is given by electromagnets, as in several solutions proposed for steering the tip of cardiovascular catheters, as well as for manipulating endoscopic capsules. Electromagnets provide an additional degree of control in varying the magnitude of the magnetic field by changing the current intensity in the coils. On the other hand, permanent magnets offer a higher volumetric magnetic flux density, typically resulting in a more compact driving system. Another interesting distinction is how to generate motion. While we have been focusing on dragging the capsule via gradient control, another possibility is to use rotating fields to advance the capsule with spiral threads rotating around its main axis. If you want to probe further on magnetically guided capsule endoscopy, then I suggest this interesting review paper appeared in Medical Physics. Now that we have discussed how magnetic actuation can enable navigation, let me give you a few examples of magnetic capsule robots that are able to perform specific functions. A first example is a biopsy capsule. The goal here is to get random biopsies in the small intestine to monitor Crohn or inflammatory bowel disease. To do this, we designed a capsule composed by two permanent magnets located at the far ends, a central unit that is free to rotate around the main axis, and a lateral opening. The central unit is composed by a semicircular blade and another two permanent magnets here, working as a magnetic torsional spring. When no external magnetic field is applied, the two magnets in the central unit rotate to expose the opposite pole to the magnet next to them. In this way, the lateral opening is closed by the back of the blade. Then, if an external magnet is brought forward, the biopsy capsule is attracted to the tissue due to the interaction with the lateral magnets. At the same time, the central unit rotates under the effect of the external magnetic field, loading the magnetic torsional spring and opening the capsule. Once the external magnet is removed, the central unit rotates back by 180 degrees under the effect of the magnetic spring, with the blade cutting the tissue and storing it inside the capsule. Here you can see a clip of the capsule in action, with opening and closing of the hole as the external magnet is moved in and out. And then, still in this video, an experiment inside explanted porcine tissue showing how the capsule is able to collect a biopsy sample successfully. It is worth mentioning that we do not have any electronically powered component in this capsule, and this allowed us to achieve extreme miniaturization and avoid limitations due to battery lifetime. Another brilliant example of smart design is the magnetic actuated soft capsule endoscope by Metin Sitti's group. This is the first example of a capsule mixing magnetic actuation with soft robotics. And also in this case, we have a bistable magnetic mechanism. When the external field is below a certain intensity, the capsule can be moved around by rolling locomotion. Then, when the operator needs to trigger a certain function, like drug delivery, the intensity of the external magnetic field can be increased to a level that aligns the capsule first, and then overcomes the resistance offered by the 4 soft hinges to the reciprocal attraction of the internal magnets. While in their first papers, this capsule was proposed for drug delivery, more recent works demonstrated needle biopsy sampling and also multi-scale robotic approach where the main capsule delivered tetherless micro grippers aiming at grasping small samples of tissue and then recovered them via a wet adhesive patch. So a combination of mesoscale and microscale robots. Another interesting example, this time mixing magnetic actuation with origami design, is a capsule robot for patching stomach wounds designed by Shuei Miyashita. In this case, a biocompatible patch is folded around the magnet and frozen in water. As the capsule is ingested and the ice melts, the patch unfolds, providing a structure for the robot to move around via magnetic manipulation and a biocompatible surface to patch a wound. So another example of magneto-mechanical design with no electronics on board. Now let's have a look at how magnetic actuation can be used effectively in different regions of the gastrointestinal tract, and in particular in the stomach and in the colon. A key element for magnetic actuation is availability of space to move in. So in the stomach, as we discussed previously, space can be created by ingesting water. Following this approach, early experiences started with manual operation of the external magnet, then moved to modified MRI machines to control the external field. And finally, the field settled on a system where a permanent magnet is controlled by a robotic arm, as in the platform developed by Ancon in China and commercialized by ANCS outside China. This platform has recently been approved by the FDA for gastroscopy and is available in hospitals in China and the US. It is interesting to mention here that the system works in open loop, without tracking the pose of the capsule and assuming that there is enough space in the stomach for the capsule to align to the external field. We will get back to this point in a few slides. Now, let's focus on the colon. Typically, the lumen is collapsed. And magnetic manipulation is not effective in a collapsed lumen, as you can see from this video. Also, the colon is permeable to water, so the same approach used in the stomach with the patient drinking clear liquid like water does not work here. So following the steer from our clinical collaborators in the Vector project, we decided to go for retrograde access, drop the wireless approach, and have a soft tether to provide gas insufflation. In addition to that, the tether provides electrical connection to the electronics and access for conventional endoscopic instruments. This design that we have called magnetic flexible endoscopes provides all the functionalities of a conventional flexible endoscope, including the possibilities to get biopsies and remove polyps. Differently from conventional endoscopes, the force to advance in the colon is applied at the tip of the instrument, pursuing a sort of front-wheel drive approach that drastically reduces the force required to progress forward. So the body of the endoscope becomes extremely flexible. This should avoid stretching the surrounding tissue, prevent pain, and eliminate the risk of perforation. Another advantage is that thanks to robotics, the procedure may become more intuitive for the operator, thus reducing the long training required to gastroenterologists. And finally, the device itself becomes extremely simple with no moving parts and a low cost of fabrication, opening to the possibility of a single-use device. So this design has the potential to address all the main limitations that we discussed earlier about colonoscopy. Of course, this is not the only robotic colonoscope that has been proposed. Other notable examples include inchworm locomotion, a crawler based on micropillar threads, and more recently, a more recent idea of self-growing soft pneumatic eversion robots. For a complete review of the field, I encourage you to have a look at this review paper appearing in the Journal of Clinical Medicine. But let's get back to the magnetic flexible endoscope. In this study performed in 2013 by Alberto Arezzo from the University of Turin, we compare robotic with conventional colonoscopy in an anatomical simulator of the lower gastrointestinal tract. To simulate lesions, pins were placed randomly and blindly to the operator. The user controlled the magnet mounted at the end effector of the robot with a joystick while looking at the image stream coming from the magnetic endoscope. The results showed that the robotic approach was comparable to conventional colonoscopy in successfully reaching the end of the colon and in the number of lesions detected. However, the robotic procedure was 3 times lower, which is not acceptable from a clinical perspective. The reason for this disappointing result was related to the assumption that the magnetic endoscope was free to move in the insufflated colon, following the motion of the external magnet. This was not always true, as sometimes the tip remained trapped into tissue folds or the tether hampered the motion of the tip in a certain direction. Since the operator was not able to recognize this immediately, the magnetic link was often lost due to the external magnet being moved away from the tip of the trapped endoscope. In these instances, the operator had to retrace the previous steps, try to re-engage the magnetic link, and move in a different direction. This was extremely time-consuming and frustrating for the operator, and clearly underlined the need for real-time localization of the magnet inside the body of the patient. This crucial need is clearly highlighted in this work by Jake Abbott's group, proposing closed-loop control of a magnetic capsule floating in liquid. Considering that magnetic field and field gradient are highly nonlinear in space, real-time tracking of position and orientation, or pose, of the permanent magnet inside the body of the patient is crucial to close the control loop. In this case, pose tracking was obtained by 2 perpendicular cameras that you see here, observing the capsule through the clear liquid. To apply a similar approach to patients, we need a pose tracking strategy that is able to localize a device inside the human body, and that is not affected by the magnetic field used for actuation. And this last point excludes all current solutions based on electromagnetic tracking, like the NDI Aurora system, as coiled sensors used there are subjected to saturation when exposed to high magnetic fields required for the actuation. So, we developed our own real-time pose detection strategy by integrating 1 accelerometer and 3 couples of Hall effect sensors inside the tip of the magnetic endoscope. The Hall effect sensors are aligned along the 3 main Cartesian axes and placed around the internal magnet. Once the offset due to the internal magnet is removed, the Hall effect sensors provide the reading of the magnetic field vector generated by the external magnet at the tip of the magnetic endoscope. This vector is then rotated by the angles provided by the accelerometer, used as an inclinometer, and then mapped in the same reference frame as the external magnet, whose pose is known. Then we perform a search in a pre-calculated magnetic field map that gives us the position that the tip should have to read these values for the external field. Since the external magnet is a cylinder magnetized along its main axis, its field presents an axial symmetry. And this greatly reduced the search space, allowing the pose tracking algorithm to run at 1 kHz. Once the pose of the internal magnet is known, it is straightforward to estimate the intermagnetic force, which is also the force that the internal magnet is exerting on the tissue in the direction of the external magnet. Monitoring this force is extremely important to make sure that it always stays below the threshold that would cause a damage to the tissue. This method was proved successful in the target workspace, with the only exception of a singularity plane that you see in this picture in the center of the external magnet and perpendicular to its main axis. There, the magnetic field is always the same, and the position of the magnetic endoscope cannot be discriminated. For this reason, we had to introduce a coil that you see here that is perpendicular to the external permanent magnet. This coil creates a field that is different at any point in the singularity plane, allowing us to identify the position of the magnetic endoscope. In this video, you can appreciate how the estimated pose is close to the true one as measured by the encoders of the robotic arm moving it around in this experiment. Of course, this is not the only possible localization strategy. There are others where sensors are worn by the patient or integrated inside the surgical bed, and others specifically designed for magnetic rolling. These are all described in this recent review paper on localization strategy for medical capsule robot, appeared in Expert Review on Medical Devices. So now that we have discussed about how to manipulate a capsule robot via magnetic field and track its pose and force in real time, the next challenge is to identify the right level of computer assistance that would make the procedure intuitive to perform for the operator. Exploring levels of computer assistance is a hot research topic in robotic surgery. Drawing inspiration from the field of automotive, this editorial appeared in 2017 in Science Robotics defined a number of levels of increasing autonomy in the field of medical robotics. We basically go from a level of no autonomy, where the robot is transparently teleoperated by the surgeon, like the Da Vinci surgical robot from Intuitive, to increased levels of computer assistance that would enable, for example, autonomous execution of tasks like suturing, and autonomous completion of parts of the surgical procedures. In our research, we have used magnetic manipulation and real-time pose and force tracking to explore how these different levels apply to robotic colonoscopy. The first level of autonomy is robot assistance. In this case, the user is controlling the tip of the endoscope with the joystick by looking directly at the endoscopic image stream. Forward, backward, left, and right are related to the reference frame of the endoscopic camera, transforming the user experience in something very similar to driving a car in a video game. The system computes in real time the position and orientation of the endoscope tip and decides how to best move the external magnet to achieve the motion desired by the user. Results from a usability trial involving 10 non-expert users, each repeating a colonoscopy in a training simulator for 5 times, showed a completion rate of 100% and a completion time of less than 4 minutes, which is comparable with conventional colonoscopy. The second level of computer assistance is task autonomy. In this case, we automated the task of retroflexion, to allow the gastroenterologist to angle the camera at 180 degrees and look behind tissue folds. This is very important because often polyps may be hidden behind tissue folds, as you can see here. A conventional colonoscope can only retroflex in the first part of the colon by being pushed against the wall of the rectum. Thanks to its enhanced flexibility, our magnetic endoscope can retroflex everywhere in the colon. However, deciding how to move the external magnet to achieve 180-degree turn is not trivial for a human operator. And therefore, we automated the task and developed an algorithm that acquires the pose of the endoscope tip in real time and computes the best trajectory for the external magnet to achieve retroflexion. In a work appeared in gastroenterology, we successfully demonstrated this approach in a porcine animal model, showing an average time for retroflexion of 11 seconds. Then we automated the task of tip levitation. In order to reduce the time that the tip is in contact with the tissue, we close the control loop in force, always applying the right amount of force that would keep the tip of the endoscope levitating in the center of the lumen. This reduced the contact between the tip and the wall from 100% to 28% of the time. This approach was improved further by applying dynamic adaptive backstepping to better reject external disturbances and enable us to perform full colonoscopies in levitation mode in training simulator as shown in this video. Another task that can be automated is ultrasound scanning. Magnetic manipulation always forces the device inside the body to stay in contact with the tissue. And this is ideal when working with ultrasound, where contact with tissue is required to guarantee a proper signal. The challenge, however, is how to control an intracorporeal ultrasound probe to maintain a stable contact during an ultrasound scan, and then how to reconstruct meaningful ultrasound images. In collaboration with the University of Glasgow, we modified the tip of our magnetic endoscope to accommodate 2 micro-ultrasound transducers, able to acquire images of the tissue with a millimeter scale resolution to provide in situ histology, and decide whether a certain lesion is malignant or not without the need for sending a biopsy sample to a pathology lab for further examination. So a first step was to define a parameter from the ultrasound signal that quantifies the quality of the echo received. Whenever this parameter was above a certain threshold, the signal was considered of good quality, as you can see here. The robotic platform was programmed to manipulate the probe against the target tissue until the echo signal was strong, and then maintain a similar strength while rejecting external disturbances, as you can see here. When performing a scan, first the platform established a strong echo, and then started the sweep in the desired direction, always maintaining a strong echo. The signal was then reconstructed by taking advantage of our real-time localization. And here we see the ultrasound image of a phantom with embedded features. Once we were satisfied with bench trial, we moved to a porcine animal model for validation. First, we reached a desired position in the column by magnetic navigation, and then we tried to acquire a micro-ultrasound image via manual teleoperation of the external magnet with very scarce results. Then we tried the autonomous routine based on real-time localization and ultrasound signal strength. And with this computer-assisted approach, we were able to acquire a clear image of the mucosa layer, demonstrating for the first time how intelligent magnetic manipulation can enable the acquisition of micro-ultrasound images from regions deep inside the body with a flexible probe. And then finally, the highest level of autonomy that we can reach, autonomous navigation. In this case, the platform autonomously recognizes the direction where to go by identifying the lumen from the endoscopic image. The green dot here represents the outcome of the lumen detection algorithm running in real time. And then the platform drives the tip of the endoscope in that direction by leveraging the information coming from the post-tracking algorithm. The only thing left to do for the user, for the operator, is to feed the tether. In recent benchtop trials involving 10 non-expert users performing 5 robotic colonoscopies each, we achieved an average time to reach the cecum of about 4 minutes, and again, 100% rate of success. This opened up the possibility for the procedure to be performed by endoscopy nurses, thus widening the availability of colorectal cancer screening for patients. We then evaluated autonomous navigation in a pig animal model, whose anatomy is more convoluted than an average human patient. And we were able to navigate autonomously up to 85 centimeters. In this plot on the left, you can appreciate how convoluted and three-dimensional was the trajectory navigated by the magnetic endoscope. confirming the great potential of intelligent magnetic navigation of capsule robots in the colon. Now, let me close my presentation with a summary of this journey and highlighting and an highlight of some of the lessons that we have learned along the way. We started from year 2000 with the introduction of wireless capsule endoscopy by given imaging. This disruptive innovation motivated the robotic community to start investigating medical capsule robots, with initial efforts aiming at providing active locomotion by integrating miniature actuators and mechanisms inside the capsule. This approach, however, struggled to provide clinically relevant solutions due to size and power constraints. So the focus of research shifted to magnetic actuation, requiring only a small permanent magnet inside the capsule robot to work. From a research angle, magnetic actuation was combined with smart mechanisms, soft robotics, and origami design. While from a clinical perspective, it reached the patient with the Ancon Navicam platform for investigation of the stomach and is now available in hospitals. What about the colon? To address the relevant clinical need of improving colonoscopy, the idea of a wireless capsule robot was abandoned in favor of a soft tethered approach, as magnetic manipulation needs insufflation to work in the colon. In the middle of the last decade, we realized how real-time post-detection was crucial to enable closed-loop control of a magnetic capsule robot in an unstructured environment such as the colon. Real-time post-tracking enabled us to explore different levels of computer assistance in robotic colonoscopy and brought us to demonstrate the feasibility of teleoperated and autonomous colonoscopy with completion times that are comparable to conventional colonoscopy. The next step for us is now to transition to first-in-human trials, thanks to the support that we have secured from Cancer Research UK and the NIH in the US. And this brings me to my last remark. As you may have noticed, research impact takes time to materialize, especially in case of clinical applications. So perseverance is extremely important. So to our young audience today, don't give up too easily. And on this note, I'd like to conclude by thanking who has been with me throughout this journey. First of all, my mentors, Paolo Dario and Arianna Menciassi, the whole Vector team, including Alberto Arezzo and Mark Schur, who gave us a strong clinical steer from the early days, and the fantastic research team at Scuola Superiore Sant'Anna, including Gastone Sciuti and Massimiliano Simi, that collaborated on many of the platforms that I have showed you today. And then my group and collaborators at Vanderbilt University and the Vanderbilt Institute for Surgery and Engineering, where I was from 2011 and 2016. Then there a particular mention to Keith Obstin, who remains one of the most committed clinical champion of magnetic colonoscopy. And of course, my current team at the University of Leeds. And last but not least, the generous sponsors from the two sides of the Atlantic that made our research possible. And with this, I thank you for your attention, and I'll be more than glad to take questions. Thank you.
Great. What a fantastic voyage indeed. Yeah, so I'd like to open up the panel here for questions again. Just for your information, the last count, we have over 3,500 people, either on Zoom or on Bilibili board. So it's a bigger audience than what can be accommodated in a typical convention center, I think. Yeah. Yeah. So we have two ways for people to ask questions. One is to raise your hand, or you can just type your questions in the Q&A board. Unfortunately, people on Bilibili cannot ask questions. So unless, okay, so there's one person who is actually raised his hand. Let me just give him the opportunity. Go ahead, this is Mr. Yeah, I can't read your name, but Kenjiro, please.
Okay. Hello, can you hear
Hello.
me?
Yes.
Okay. Thank you very much for your very nice talk, Professor Pietro Valdastri. I'm Kenjiro Tadakuma, mechanism guy in Tohoku University, Japan. And my question is, in the future, of course, it's not so easy, but do you think it is possible to realize the robotic capsule or colonoscopy just only with biocompatible or degradable materials? It's like a sort of the whole body biodegradability and so on. But
Yeah, so I think biodegradable materials are extremely interesting. I can see them more as something for therapy and drug delivery. So I think biodegradable material loaded with drugs can be extremely interesting to navigate to a specific point, remain there, and get dissolved. Very similar to what Shuei Miyashita did, and I presented for that patch to close a stomach wound. So the application really for guiding something to a specific point so that it can stay there and degrade. Overall, biodegradable material, I see difficult to, at the moment, create a camera that is biodegradable, and also the electronics. I think that's the main challenge that I see.
Okay. So from the actuation, it's not so easy, but yeah, I really hope to see the next.
Yeah.
Okay, thank you again. Thank you very much for your very impressive talk, and thank you, and thank you, Professor Hon too.
Sure. Yeah. So yeah, let's move on to maybe a question in the Q&A board. So the first question is from Jay Yang. The question says, "Hi, Dr. Valdastri. I would like to ask if the robot is powered by electromagnetic field. Does it friendly, or I guess is it friendly to the patients who did stenting surgery before?"
Yeah, that's a very good question, and that one that I get very often. So at the moment, we see the same limitation that magnetic resonance imaging has. So we don't see this technique to be applied straightforwardly to patients with implants that are susceptible to magnetic fields. So if the stent is as non-magnetic material, then it's fine. But if the stent may interfere with the magnetic field, then probably is an exclusion criteria. Nevertheless, the magnetic fields that we are using for moving the capsule inside the body is so localized that probably this restriction can be lifted in the future. For example, especially if there are no magnetic implants in the abdominal area, then the procedure can go ahead because it's very, very limited, the range of the magnetic field, and the field drops so fast with distance that the workspace is very confined.
Okay, so thank you. So the next question is from Cameron Forberger again. And the question says, "It seems that your lab is developing many practical devices that are near to the point of commercial application. What is your strategy philosophy when it comes to intellectual property?"
Well, in my lab, what we implement is the following. So basically, whenever we are doing something relevant, very new, at the time we submit the paper to a journal for evaluation, we submit the same paper draft to our technology transfer office to protect the IP. And so, for example, the localization technology that I've shown, that's a big core of the IP that we have. And if we are successful with first in human trial, then will be the core of a startup that will commercialize the platform. So the short answer is, At the time we submit a paper, we submit the same to the Technology Transfer Office of the university, and fingers crossed they are supporting us with filing a patent.
Right. Okay, good. Okay, the next question in the board is from Zheng Li. Great talk, thanks. Is there any barriers do you foresee for magnetic colonoscopy applied to or on humans? What's the clinician's comment comparing to the conventional colonoscopy and magnetic flexible colonoscopy?
Yeah, so very good question. So the main obstacle that we had to address was time. So time to get to visualize the entire colon, to get to the end. And we solved that issue with real-time localization and intelligent control. So now we are able to get to the end in 4 minutes, which is comparable with conventional colonoscopy. And now we believe we are at a point where we can compare 1 by 1 with conventional colonoscopy, and that's why we're moving forward to human trials. I believe, we believe that magnetic colonoscopy will be painless, completely painless, so not requiring sedation. It will be easier for the operator, and then the device itself can be extremely low cost. So part cost is about $50. So hopefully, we hope that it will replace colonoscopy eventually, but certainly if not replace, it will be an option for a colonoscopy outside the hospital setting. So performed by non-expert gastroenterologists outside the setting where there are cleaning reprocessing facilities that are not needed in case the probe is disposable. So our main goal is really to enlarge the number of screening procedures and save lives.
Okay, great. Maybe 1 minute. Maybe I can sneak in a question myself. I don't work in this field, but I am sort of familiar with the research. Do you have malfunction happening from time to time? And if it does happen, like the capsule gets stuck, what's your contingency plan normally? So
specifically for the colonoscopy platform, the contingency plan is the tether. So since it is not wireless, it has a tether, you can just pull the tether and you retrieve the capsule. So that's also safety, a big safety item. In case of a wireless capsule, then the idea is that it goes down with peristalsis. So eventually it gets expelled by the body. And that's the same contingency plan that given imaging is applying. It's very small retention. As long as the size is small, retention is minimal.
Good. Okay, I think that's all the time we have. So thank you again for a great talk. People online, please join me in thanking Professor Valdastri for a great talk. Okay, thank you.
Thank you.